So this is Shubham, Shan, telling us that zebrafish are in fact awesome. I bet you didn't know that. Well, now you do.
Let's see.
Click allow.
It works! Woo! Okay. Okay, so just a show of hands, how many people know what zebrafish are? Whoa, look at you guys. Good. Okay, so who am I? So my name is Shubham, third-year honors physiology. So my name is Shubham, third-year honors physiology student at the University of Alberta, and just have some. I didn't realize that is a little creepier in retrospection. And that's me. Yay. Okay. So before we get started, what exactly are zebrafish? So I'm not going to go into genetics of it, but zebrafish are basically these tiny fish that were first discovered or found in the Himalayan regions, and but they've also been found now in the Caribbean. Many people might have them as pets. What are the positives of using zebrafish in research? So they actually have a very fast generation time. So what that means is that by 48 hours post-fertilization, you can basically use the fish for research because it's an independent functioning organism at that point. It also is easy to care for compared to a mouse, a model that you may use for research. And they're also transparent. So here's just a list of things that you can use it for, and the list is exhaustive that you can use zebrafish for. That's an adorable little zebrafish. And these are so the only negative for zebrafish is that it's not still a perfect mammalian model. So what that means is that when looking at a human disease, the most preferred model that you use for research is a mammalian model, like a mouse or a rat. So you can actually have research studies that won't get funded because of the fact that you're using zebrafish. So this is, just ignore everything else, just look at this part and that part. So the 16-cell stage of the zebrafish is actually ready to go by 2 hours post-fertilization. And that's how quick they develop. And by the end of 2 days, you actually have, like I said, an independent organism. So you can do tests throughout the period of development of zebrafish, and that's not a full fish, of course. That's a juvenile zebrafish. But that's a 48-hour zebrafish. So most of my studies that I have done have been around this. But you modulate characteristics initially so you can measure things there. So what are my goals for the next 10-ish minutes if we don't get kicked out or wrapped up? So I just wanted to just talk about what research that I've done at the university. And I basically worked with fetal alcoholic spectrum disorder, which is different than fetal alcoholic syndrome disorder, and I'll get to that soon too. And I'm just going to briefly talk about auditory and visual studies that zebrafish are useful in. So everyone, I'm sure, knows about ethanol. So ethanol has been intimately related with human history throughout fermented rice, wine, and all of that fun stuff. And ethanol, basically what it does for the body is that it accentuates or it increases the inhibitory mechanisms, like GABA, and it depresses the excitatory mechanisms of the body. So, and it's the most claim to fame for ethanol is when it's used with other drugs, I guess, that you see on the news. An example would be Michael Jackson, who took propofol and ethanol and choked on his vomit. So that was fun. And that is the most common cause of medical emergencies. So with zebrafish and ethanol studies, what I was responsible for, what I did was I basically immersed fish, the new eggs that we got, and I immersed them in different ethanol concentrations that had varying concentrations. And what those concentrations I immersed them were during the gastrulation period. So I'll come back to the slide. But the gastrulation period, yay, bio-30. So gastrulation period.
I'm B.Sc, we don't believe in science, so stop it.
Okay. So gastrulation is basically when you have the embryo that basically forms 2 germ layers, so the endoderm and the ectoderm. And basically, generally, ectoderm gives you your neuroectoderm, so your nervous system, and surface ectoderm, which is your skin. And the endoderm is all the inside stuff, and then later, everything in between, the mesoderm forms later on. So I'll come back here now. So that's why we chose the gastrulation period, is because most critical initial processes that happen in development happen during gastrulation. So we wanted to see what would happen if you expose the fish to ethanol during that period. and then measure various responses at 2 dpf, like morphology, behavior, and immunohistochemistry, which sounds like a big word, but I'll come back to it too. So as to what it looks like in real life. Real life. This cartoon, that's real life. Just in case you were confused. So this here is basically invagination. So, yay, science makes sense. So it's right there. Like once again. Okay. So, we did 4 concentrations. We did 0 micromolars, 10 and 50, and 100 micromolars. I'm not going to show you everything, of course. I'm going to leave something for the research paper. So here I have just 2 extremes, of course. So this is a control, and this is 100. And you might say, oh, they look so similar. And yes, they do. And it will make sense once we go ahead. So this normal eyes, normal yolk sac, tail, yeah, it's happy. And so is this one. Well, there's this, I guess, a little edema around the pericardial edema, which basically prevents the heart from beating, efficiently at least. No, they just swim around and they're adorable and everything. But anyways, we'll go to, this is really cool, so movement disorders. So this is, we did this really hard, I did this really hard technique. We basically took zebrafish embryos. So these are that have been treated at 0 or 100 for, I guess, 2 days. And they've been treated for a period of 5 hours and were looked at 2 days, sorry. So what you do is you take the fish, you put it in a petri dish, and then you take low molecular, we got ethics approval for this. You take low molecular weight agarose, and you put the agar on top of the fish, and the agar solidifies, and then the fish is under the agar, and then, this is really cool, because I came up with this, and then you cut out the tail of the fish, right? So the tail can be moved. So you can measure tail curvature, tail radii, all that physics stuff, which I don't know. And we have a statistician in our lab that does this. And this is a pipette. So you take the pipette and you place it next to the otolith, which is the zebrafish's ear, per se. And you will, I guess, spritz out, or whatever the appropriate word is, onto the otolith some liquid at a certain pressure and a certain volume. And then you look at the tail. So you see, let's see it in action. So quite normal, this is control, so quite normal for, you see good tail curvature. So you have an escape response, which is the initial response that you see, and then you have general swimming behavior. So as a response to an auditory stimulus, you have an escape response and general swimming behavior. So this is quite normal. Despite having, and just keep in mind that the ethanol and the control have very similar morphologies. So they look the same outside, but inside there's some crap going on. Okay, so now I'll go to it's just doing its thing. I think it's a pretty long one. Okay, well, I'll go to the next one. So this is ethanol one. So it's trying. So not only can you see an initial delay of movement, but then you also see general coordinated movement. And now why this is important is because now if you think about fetal alcohol syndrome and fetal alcohol spectrum disorder, fetal alcohol syndrome is when you have very abnormal, very intensely abnormal physical, morphological, and behavioral characteristics. But that is not what is common these days, I guess, in the female population who are pregnant. When I guess humans, when we drink, we actually don't drink everything at once. We binge drink, especially when you're pregnant, you don't know it. So this is a very low concentration. Now, this sounds like a really high concentration, but that would, I think I calculated it out once, it would be like quarter of a shot and of some alcohol, vodka, I think. But anyway, so you look at the movement, and then you see, even with that movement, over treatment of 5 hours, you see a very, I guess, adverse effect on movement. And now the cool stuff, the immunohistochemistry. So this is basically you tag things, you tag proteins, and then you're like, oh, this protein is expressed here, and how well it's expressed, and how well neurons are expressed. So this is a control. This light should be turned off, but we'll deal. So these are the Mothner. Oh, yes. Who was it? Jesus. Woo! Okay, thanks, whoever was that, Jesus. So this is the fish's head. So what we did, we tagged neurons with certain fluorescence markers, and you see this is the fish's eye there, and these are M cells, with Mothner cells, and these are the otolith cells. So you can see, look pretty normal, round with normal, and this is actually a single neuron. So you learn about neurons, and you can see neurons. So this is exciting a little. So M cells and otolith cells, and just keep this in mind, and I'll go to the next one. Party. You can see abnormal otolith cells, so abnormal sensing mechanisms for auditory stimuli. And you have these Mothner cells. If you go back to this, you see how well displaced with a higher volume per ratio of cell that you have there. Here you have a lower cell. So there's not only is there otolith stimulus, but if we had tested for some sort of visual defect, we would have seen crap. Okay. Okay, and the eyes are abnormal too. Yay. So now, I'm not going to go too much into sensory because I don't have time, but sensory organization in humans is very complex. So the visual layer, for example, has 10 layers of repetitive neurons that are receptive neurons, in the retina where light strikes and then you see and things are complicated. And in the auditory, you have, just read that, medium change derivation of signals converging to mechanical hair stimulation in liquid. So that basically means that hearing is hard. And this is basically you take vascular endothelium or vascular cells in zebrafish. So these are all blood vessels that you can see in zebrafish. And you can actually use them for modeling stroke, or you can use them for modeling diabetic retinal myopathy, where basically due to hypertension you get diseases in the retina. And this is another cool picture where you can actually take the layer of retinal cells, and then you can treat it with the genetic component, you can treat it with ethanol, you can treat it with anything you want, and then observe visual defects in response to that. And in conclusion, so zebrafish, there's many spheres of research. Some of the research that is happening at the U of A, so you have retinal stem cell regeneration therapy. Dr. Ted Allison, leukemia is pretty new actually for zebrafish. There's actually cardiovascular leukemia oncogenes. Dr. Vaskowicz, FASD, which is what I did. And there's quite new to the kidney transport disorders. That guy, Zheng Chen. And then, so zebrafish overall is very versatile. And this is the coolest thing I did this summer. Wait for it. Woo. So, yes. Okay. So you can basically take single neurons. You can basically take cerebellar neurons, and these are iris. Basically, the picture I showed you before on a smaller magnified scale. You have the spinal cord. You can take primary sensory dorsal ganglia. You can take the otolith cells. You can take lots of stuff, and then you can look at multifactorial organ association therapies. Basically, that sounds like a big word. And you don't need to know that. Questions?
So a couple questions. Who has one? Sarah.
Do you have like a dental retirement program for all your test fish afterwards?
Yeah, we just drug them. Sorry. So according to protocol, if this adult fish, you decapitate them or you put them in TTX, which is tetrodotoxin, so it blocks muscular, it's basically death row. You do like 3 different compounds. So you stop them from moving first, then you give them something that stops their heart, and then they die. And then with the larvae, you can just flush them down the sink. Proper lab techniques.
So that last picture you showed, so is that after they're already dead, or can you do this?
Oh,
in
dead.
real-
Yeah.
Can be done in real time?
No. So what the process of fixing fish for immunohistochemistry, which is what this is you put them in formaldehyde.
Why use GFPs?
Well, you can use GFPs, but that's not what I did. You can use GFPs. Grain fluorescence protein, you can use them naturally expressed in fish, so you can do that for sure. But I wanted to look at more than one thing. We want to look at more than one thing. GFP, when you use GFP, you have to actually use different genes to target different neurons.
You bet, but then they're alive.
And you
Oh,
can watch it in real time.
that's exciting. Not much fun, though.
We'll do one more question, if there is one, from not over there. Yes.
When are you starting to learn about the alcohol spectrum
Disorder. So this is I worked on this last summer, and we basically learned that, so I didn't go into much detail about it, but the gastrulation period of zebrafish actually corresponds perfectly with the gastrulation period of humans in the time scale at least. So zebrafish gastrulation period is 5 hours post-fertilization to 10.75 hours post-fertilization. That's the time we incubated them in ethanol for. But in humans, the period corresponds in the sense that most drinking, it's a social aspect to it, most drinking of females that are pregnant, but they don't know it, is during that initial phase, which is around day 4 to day 5. That's if your ovulation cycle is day 14, you get fertilized 15. That's 5 days, 20. So before you even get your period, well, not you, but before they even get their period, it's basically by day 20, By day 20, it's still not with their cycle. So they might think that they are not pregnant because they don't have the period, so they will start drinking. And that's where you see these defects. So spectrum disorder, anyone here might actually have fetal alcoholic spectrum disorder. You don't know because they look normal, but they might have slightly variable motor. That's the whole point, is to see that even if little amounts of alcohol can actually affect movement disorders in humans, based on the fact that you have a totally, I guess, one end of the spectrum is where you have clearly, well, not clearly, you have...
Bad at sports?
Maybe. Probably. So you can look at two extreme spectrums. So you can look at someone that's normal and expresses some symptoms which are may be noticeable, may not be noticeable, versus someone that is clearly mentally, physically, and morphologically retarded.
So can that be tested, the spectrum disorder?
I don't know. Fetal alcoholic spectrum disorder is quite new from what I know. So it can be tested, but how do you test it is a question. So you can look at general variations in motor function. For that, you need a control-sized population. So you have to get a control population, measure their control function, and then see if people fall in statistics, so in some standard deviation from that control population, if that makes sense.
All right, thank you, Shubham.